Yeah. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Distinguished speaker, Professor Vivi Kasim, PhD. Distinguished moderator for our guest lecture, Professor Dr. Rare Physiology Septelia Inawati Wanandi. Distinguished head of the doctoral program in Biomedical Sciences, Professor Dr. Rare Nat Asmarina, MS, and also Professor Wu from College of Bioengineering, Chongqing University, which has been from Professor Vivi. Hello, Prof. Okay, thank you so much for coming. And also the audience, beloved students, offline and online in this session, and all the esteemed guests that here for our today event guest lecture. It's a great honor and pleasure for me to welcome you all to today's guest lecture organized by the doctoral program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia. And today we are privileged to have a speaker, Professor Vivi Kasim from College of Bioengineering, Chongqing University, China, who will deliver a lecture entitled Chromosomal Instability in Cancer, Paradox and its Application for Overcoming Immunotherapy Resistance. In this session, we will hope we can get a lot of insight and foster a future research. In this guest lecture, you can also give questions about the research opportunity in the future in the Chongqing University as well. So without further ado, let us begin our today's event by inviting the head of the doctoral program in Biomedical Sciences to deliver the opening remarks, to the respected head of the doctoral program in Biomedical Sciences, Professor Asmarina. The time is yours.
Okay, good afternoon, yeah, almost afternoon, yeah, good morning and almost afternoon. Our guests, of course, and our student here, our guests, our participant. Of course, I'm Asmarina as a head of the doctoral program in Biomedical Sciences. Would like to sincere thanks to Professor Vivi who want to deliver her expertise in the cancer, yeah. And it is a great honor for us to have you today to speak or to explain about the topic of the chromosomal instability in cancer, paradox and its application for overcoming immunotherapy resistance. Yeah, this is the challenge, interesting topic in cancer, yeah. And thank you very much for this opportunity. And as I know that you will also deliver about the opportunity to do research in Chongqing University. And I think this opportunity also good for us, not only for the student that may continue research for PhD thesis or thesis or research in master degree, but also for our lecturer, because you said also there is also opportunity postdoctoral research for our lecturer in our faculty. So of course, our doctoral program will conduct. Our study program encourage the student that can supervised by foreign supervisor, because we think that the international experience to do research in the abroad institution is good for our student. So and therefore, I would like also thank to you that you also your willingness to be supervised for our student, yeah, because one of our student also supervised by you, yeah, in our for the dissertation or thesis research. So, and for the student, I would like to say that please use this opportunity to get knowledge, yeah, recent knowledge and sophisticated knowledge maybe about the cancer. And also the opportunity maybe to conduct research in Chongqing University in China. And Professor Firfi told us that there is also funding for that, yeah, from the China government to do to conduct research in Chongqing University. So thank you very much, and thank you also for Professor Wu has already accompanied your wife to be here, yeah, to this guest lecture. So yeah, I think we open this guest lecture from Professor Vivi Kasim, and thank you also for the student or the lecturer here. There is lecturer, no, yeah, for coming in this guest lecture, and also for the participant in Zoom. Yeah, how many participants now in the Zoom? Any link Zoom? 26. 26 participants in Zoom. Yeah, yeah. Okay, thank you. Maybe it can be also more and more. Yeah, and during the time. Okay, thank you very much, and yeah, please enjoy this guest lecture from Professor Sisi Kasim. Yeah, thank you.
Okay, maybe as applause for Professor Asmarina. Okay, thank you so much, Professor Asmarina, for the opening remarks. Also, we hope these today's lectures will be provided our knowledge more and inspiration also for us. So, ladies and gentlemen, we now come to the main session of today's event. But before we begin, it's my pleasure to introduce our moderator for this session, Professor Dr. Rare Physiology, Dr. Saptelia Inawati Wanandi, who will guide us throughout the lecture and discussion session. Prof. Ina, familiarly we call Prof. Ina, okay, is a professor from Biochemistry and Molecular Biology Department, Faculty of Medicine, Universitas Indonesia. She also head of Molecular Biology and Proteomic Core Facilities, IMERI, Faculty of Medicine, Universitas Indonesia. She also researcher on the Mandaya Royal Hospital Puri research team, which is there is Pak Edi in here, the director of the Mandaya Royal Hospital Puri. Good afternoon, Pak Edi. Okay, so Prof. Ina graduated from Medical Doctor, Universitas Indonesia, and continue her study in Magister program, specifically in Human Biology in Philips Marburg University, Germany, and continue Doctoral program as the same topic, Human Biology, in Philips Marburg University, Germany. So without further delay, I would like to invite Professor Ina to take the stage and lead the session. To Professor Ina, the time is yours. Applause, Professor Ina. Maybe you can use this.
Okay, thank you very much, Nusli. So, good afternoon, ladies and gentlemen, especially distinguished Professor Vivi Kasim and Professor Wu, when I'm not mistaken to spell this name. And also students here. And we have 26 participants, 26 participants in Zoom. Good afternoon. Maybe doctor and professor in Zoom. Okay, now I would like to ask Prof. Vivi to come in front. May I? So please come in front. I will introduce Prof. Vivi. Prof. Fifi Kasim is currently a professor and principal investigator in the College of Bioengineering, Chongqing University, China. She completed her bachelor and master degree in chemistry and biotechnology from the University of Tokyo, Japan, before earning her PhD in the same field for the University of Tokyo, Japan in 2007. From 2007 to 2009, she held a Japan Society for the Promotion of Science, JSPS, postdoctoral fellowship at the Institute of Molecular and Cellular Biosciences, now the Institute of Quantitative Biosciences, the name. The University of Tokyo, in the University of Tokyo. She then served as research resident at the National Cancer Center Research Institute, Japan, from 2009 to 2011. In 2012, Dr. Kasim joined Chongqing University and established her independent research group together with Professor Wu, her husband. Her team focused on reciprocal regulation between tumor microenvironment and tumorigenesis, chromosomal instability and tumor drug resistance or drug persistence, cancer stem cell population maintenance, development of small molecular drugs for tumor and diabetic food from natural product and traditional medicine, development of stem cell-based and gene-based therapy for tissue regeneration in diabetic field. Prof. Fifi has published over 60 peer-reviewed papers in top-tier journals, including Science Advances, Signal Transduction and Targeted Therapy, Cancer Research, Advanced Science, and Oncogene. She also served as Associate Editor of Critical Reviews in Oncogenesis and Editorial Board Member of BMC Medicine. At the moment, Prof. Fifi and I supervise a PhD student in Doctoral Science of Biomedical Sciences. Please stand up. So Dewi Hammer is the student supervised by Prof. Fifi and I. And we also obtained the joint supervision program from our university. So Prof. Fifi, your time is 1 hour, then will be followed by a question and answer. So time is yours, please.
Okay. First of all, I would like to say my appreciation to Prof. Ina and Prof. Asmarina for this wonderful opportunity. So as mentioned by Prof. Ina before, I have the opportunity to supervise one PhD student from FKUI, Dewi. And we have obtained recently joint supervision grant, and hopefully we can start more specific and more collaboration. So because there are also some other opportunities for collaboration. For example, joint grants from BRIN in Indonesia or LPDP with the NSFC from China, and also hopefully in the future, in the near future, we can start a joint lab, which is also a very pushed program by Chinese central and also local government and our university. Okay, so actually I planned to deliver this topic in Indonesian, but as I see there are also international students, so I changed to English. But for the questions, I welcome both questions in Indonesian and in English, so please feel free. Okay, so my topics today is the chromosomal instability in cancer paradox and its application for overcoming immunotherapy resistance. Okay. So first of all, maybe I would like to introduce you. I would like to spend several minutes to introduce you Chongqing. This is a city which is now viral in the internet. So, yeah, several years ago, if I say Chongqing, maybe people don't know about this city. Famous city from China is, for example, Beijing, Shanghai, but Chongqing, maybe several years ago, nobody knows that here. But nowadays, there are many tourists from Indonesia to Chongqing because it's very viral in the net. So, this is a city with a more than 3,000 years history, located at the joint of two big rivers in China, Yangtze River and the Jialing River. It is located in western China. It covers more than 82,000 square kilometers with a population more than 30 million. This is the youngest of the 4 municipalities in China directly under the central government. The other 3 are Beijing, Shanghai, and Tianjin. So this is a mountainous city. So actually, the tag for Chongqing, the classical tag for Chongqing is mountain city, bridge city capital, fog city, and hot pot. So you have a lot of hot pot here. So I even found a hot pot restaurant original from Chongqing in a mall in Jakarta. I forgot Taman Anggrek or whatever. But yeah, and also the mala, the spicy mala taste. Yeah, this is like the spice from Indonesia, andaliman also, right? So this is also from Chongqing. So why people in Chongqing like to eat hot pot and mala? Because Chongqing is surrounded by mountain and it has 2 big rivers. It is very humid karena uap airnya nggak bisa naik. So it's very humid. So people eat hot pot. Yeah, it's also foggy. People eat hot pot and mala to help them to get warm and supaya keringetan. Karena nggak gampang berkeringat, itu susah. So Chongqing also is actually a very important traffic line in China because there is a very long railway starting from Chongqing. This was established in 2011, if I'm not wrong. From Chongqing to Xi'an and then Xinjiang in China, then goes to Kazakhstan, Russia, and then Istanbul, Turkey, and Poland, and Belarus, and ended in Germany. And some of the branch of this railway also reached Rotterdam and Antwerp in Belgium. So this is a very important railway line so that the product from China and also from Europe do not need to go shipping. So it is a very important economic railway line nowadays. And Chongqing, you can see here, this is the viral one in the net. So the first one, this is a steel architecture. Said it is like the architecture in the Japan animation, Sen and Chihiro. And this is the one very viral, the train that goes through the building. And also, this is Three Gorges Dam, the biggest dam in the world. It is also under the municipality of Chongqing. And you can see the flyover. It is very confusing. And it is said that GPS is beaten by this. You will get lost even if you use GPS. Yeah. So yeah, there is now direct flight from Jakarta to Chongqing 4 times a week, starting from last November, I think. So how about our university? Chongqing University, because most of Indonesian students, if they study in China, they go to Beijing, Shanghai, or Shenzhen, or Guangzhou. But actually, Chongqing University is top 35 university in China. The rank is 30-something. And the international ranking, our international ranking actually is quite high for research If we just take a look on the indicator of research in QS, it should be around 150 in the world. But the problem is the internationalization, so it makes our rank, yeah. But for example, for the US News rank, during 5 years, within 5 years, our university's ranks lived for 187, nearly 200 ranks. So it's about 200 something nowadays. So Chongqing University was established in 1929 and is directly under the Ministry of Education of China. This is a key construction university under the State 211 project, which includes top 150 universities in China and 985 project, or top 39 universities in China. We have now about 50,000 students. And also we have 7 faculties, with the Faculty of Medicine re-established, because we have it previously, but it disappeared. But now we re-established the Faculty of Medicine, which includes our College of Bioengineering. And we have about more than 5,000 faculty, 5 plus N campuses. I don't know how exactly the number is, because we are now building a lot of new campuses and research core facilities. So I'm not sure about the number right now. So our college is College of Bioengineering. We have also a platform from the Ministry of Education, the key laboratory of bioradiological science and technology. And the platform, another important platform, is the 111 Project Laboratory. So these are some photos from our international students' activity. This is the welcoming party and orientation for international students. So that is a glance about the city and the university and the college. So let me start my topics today. So as we know, in human cells, we have 46 chromosomes. And abnormalities of the number and structures of chromosomes will lead to severe diseases. The most famous one is the trisomy: Down syndrome, Patau syndrome, and Edwards syndrome. So we know that the chromosomes should be passed to the next generation, either as a species or a cell, in a very exact number and structure. And we have a lot of mechanisms to guarantee this during cell cycle. But there is a very interesting thing. We also know that in tumor cells, actually in more than 90% of tumor cells, the number and the structure of the chromosome is not normal. So we can see this is the normal human chromosome here. But in the tumor cells, you can see the number and also the structure. There are a lot of recombinations, right? And also, summarized by Dr. Hanahan and the Hallmarks of Cancer, the latest one, published in 2021, genome instability and mutation is one of the 14 hallmarks of cancer. So this is strange. We know that it is not allowed, it cannot be tolerated by normal cells to have abnormal chromosome number and structure, but tumor cells have it. And actually, it is said that in tumor cells, actually this abnormalities or chromosomal instability, or we know it as CIN, is one of the driver of tumorigenesis. So tumor cells not only can tolerate it, but actually it drives the progression of tumor. Why this happened? Because you can see here, by gaining the chromosomal instability, for example, having another copy of one chromosome, one specific chromosome, will make the tumor cells becomes more adaptable to the circumstances. And it increased the heterogeneity in the tumor tissues, genetic heterogeneity in the tumor tissues. So for example, this is a very good example. So in some of the tumor cells, of the clone of the tumor cells, they observed that the MAPK13 increased. And MAPK13 is in the upper of the ABCG2. So the copy number of MAPK13 increased. The chromosome that brings this gene, they have another copy, more than 2 copies. So by that, MAPK13 expression increased, and this will increase the expression of its downstream target, ABCG2. And ABCG2 is a drug efflux. It's a drug pump. So by this, the tumors, these clones can pump out the drug, so they become more resistant. And for some clones, they might have more copy numbers of gene related with metabolism, or gene related with hypoxia or other characteristic of tumor cells to adapt to the environment. So it seems that the CIN will benefit the cancer cells, right? But there is another important thing from the clinical patient data. So this was reported in Cancer Research, and after that I will show you that our analysis get the same conclusion. So if we stratified the clinical patient cancer into low CIN, so we stratified them into 4 groups, 25%, 50%, 75%, and then the highest one. There is a very interesting phenomena. The group with moderate CIN has the poorest prognosis, but the group with higher CIN has the best prognosis. So this is confusing, right? Because here we say that, okay, CIN can increase tumor heterogeneity. It drives the tumor clonal evolution to select the cells and clones that are more adaptive to the microenvironment, and this drives tumor progression. But patients with a higher CIN has the best prognosis. So this leads to the establishment of a famous model in CIN, which is called the just right model of CIN in cancer. In this model, it is said that, that the CIN, in a certain degree of CIN, can support tumorigenesis because it increases the adaptability of tumor cells to the environment and so on. But if it exceeds the threshold, then it will cause tumor cell death. However, this model lacks experimental evidence. And so the role of CIN in determining tumor cell fate was still unclear. Because people usually do the experiments or research about the low CIN or very high CIN, no research that can focus on the whole degree of the CIN. So we started these topics about 8 years ago. So there are 2 hours of our PhD students who accomplished this research. One is Randy Hosea, who graduated from FKUI for his master degree student and a student of Prof. Ina. So yeah, he started a lot of experiments here, and he did it in very diligently and hardworking. So we, by using TCGA database, we tried to analyze what is the reason of the CIN, what is the major reason of CIN in tumor. And we found that the chromosome missegregation via mitosis errors or telomere dysfunction is the main cause of mitotic errors in tumor cells. Because we know that the chromosome number becomes abnormal, the problem should be in mitosis, right? The cell division. So the main problem should be in the missegregation. We know that sister chromatid needs to segregate and move to the 2 poles of the cells during mitosis. This should be, the abnormality in this point should be the main cause. So to prove this, we collect the cells from each phase of the cell cycle, the G1, S, G2, and M, and perform the transcriptomic analysis. So this is the DEGs from M versus G1, M versus S, and then we cross the DEGs from these 2. So we have 3 samples. We compared first M and G1, and then M and S, and we got the differentially expressed genes or the DEGs, and we crossed them. So we got the DEG specific for M phase. And then we try to search for transcription factors because transcription factors can regulate a lot of genes at once. And there is a gene named YY2. This gene is differentially expressed in M phase cells. This is a transcription factor. Sorry. Oh, okay. Okay. So let me introduce you. This is a YY family, yin-yang family. Don't think about the martial arts, kung fu, no. This is just the name because this yin-yang, these transcription factors have both roles as a transcription activator and suppressor, plus and minus. So it fit with the ancient Chinese philosophy, yin and yang, plus and minus. Day and night, dark and light, and so on. So the professor who found this factor named it yin-yang family. So YY1, the member of this family is YY1 and YY2. They are zinc finger protein transcription factors. And YY1 is conserved in the animal cells and some of the plant cells. But YY2, it is only conserved in placental mammals. Because it is said that YY2 is originated from YY1 mRNA, which was retransported and inserted between exon 5 and 6 of another gene during the evolutionary time after the divergence of placental mammals. So it is only conserved in placental mammals. The homology between these two transcription factors in zinc finger domains is over 80%. Okay, so we confirm the expression level of YY2 during cell cycle. So we synchronize the cells with serum starvation. By this, cells are synchronized in the G1 phase, and then after the release, after we added the serum again, then after that, cell will enter the cell cycle at the 16 hours after adding the serum, or after the release from starvation. Cells will enter the S phase, and after 20 to 24 hours, they are in the M phase, actually. So during this cell cycle, you can see that YY2 overexpression increased and peaked at M phase. So this is a time-lapse microscopic images. As you can see here, the mitotic time, or the time between, the first event of the mitosis is the nuclear envelope breakdown until the end of anaphase. We measured this time. The time of the mitosis for control cells is about 50 minutes, 50-something minutes. But if we overexpress YY2, then the time was prolonged until more than 70 minutes. And it is because the increase of the spindle assembly checkpoint activity. Spindle assembly checkpoint is one of the very important checkpoints during cell cycle that check whether sister chromatids already line up in the equatorial or not. If the sister chromatids are not lined up yet, they will not allow the sister chromatid to segregate and move to the two poles of the cells. So because YY2 enhances this SAC activity, then sister chromatids cannot be segregated and moves on. So this is the point. Sorry, would you please can you please play the video here, or no? It's okay then. It's okay, yeah. So, and actually, our lab has focused on YY1 and YY2 for several years, and we know that YY2 is a tumor suppressor. So when we overexpress YY2, cell death increases, and then also this is the EDU incorporation assay. So cell proliferation decreased, and also cell viability decreased. But if we treated the cells with reversine, which is an inhibitor of SAC activity, then cell death decreased, cell proliferation increased, cell viability increased again. So it seems that the inhibition of SAC activity cancels YY2 tumor suppressive effect. So I'll skip the detail for the mechanistic study, but through the cross-omics analysis using the transcriptomic analysis and also this one, cross-analysis using RNA sequencing, ChIP-seq, and so on, we actually obtained the molecular mechanism. That is YY2 increased the transcription of BUB3. This is a component of SAC complex, spindle assembly checkpoint complex. So by that, because YY2 increased the expression of BUB3, it caused the SAC hyperactivation, and then cells cannot move from metaphase to anaphase, and it prolongs the mitosis. What happened is, actually, even if the mitosis was prolonged, the cells will divide eventually. But during that division, the chromosome will be segregated not normally, and it will cause chromosome missegregation. So I said that it is just like a ziplock, kayak sleting. If we pull down neatly, it will be very fine. But if you shear up, then it will be broken, right? Seperti sleting, kalau kita buka secara benar, itu kan terbukanya rapi ya, nggak masalah. Tapi kalau dipaksa, robek. Itu yang terjadi kalau menurut saya, seperti itu imagine-nya. Okay, so now, what is the effect of YY2 on CIN? This is confusing, honestly saying. So first, if we knock down YY2, let's see, this is the DNA content. So here, 2N, here is 4N. We know that our chromosome, we have two sets of chromosome or 2N, right? So after S phase, before the cells divide completely, then we have 4N because the chromosome has been copied during S phase. In control cells, this is tumor cells, but it is CIN negative. It is said as CIN negative. You can say that the percentage of cells with 5 to 8 N of chromosome was very low, less than 2%. But if we knock out YY2, you can see here the percentage of cells with 5 to 8 sets of chromosome increased drastically. Okay, but you can imagine this because the SAC activity become very low. Then the sister chromatid will be segregated and move to two poles before the time, right? But the problem is when we overexpress YY2, the same condition occur. The 5 to 8 cells with 5 to 8 sets of chromosome also increased. So usually, knockdown and overexpression, or knockout and overexpression, you will get the contrast phenotypes. But we got the same things. And this is the karyotyping. Oh, sorry. Yeah, this is the karyotype. The karyotyping. So we counted the number of the chromosome. So HCT116 is a colorectal cancer cells from a male. It has 45 chromosomes because it lost its chromosome Y. So how about the number of cells with more than 47 chromosomes? Almost no that kind of cells in the control group. But when we knock out YY2, the percentage of cells with more than 47 chromosomes increased to more than 30%. But when we overexpress YY2, again, it increased. It's paradoxical, right? Confusing. So another indicator of CIN, chromosome bridge. You can see that sister chromatid here already reached the two poles. But here, you have sister chromatids still linked together. This is what we call the chromosome bridge. The number of cells with chromosome bridge also increased in both YY2 knockout and overexpressed cells. So this chromosome bridge actually will cause another indicator of CIN called the micronucleus. So this chromosome bridge will eventually broken up, and the chromosome will be enclosed by a membrane structure like a nucleus, but this nucleus-like structure is in the cytoplasm. We call this micronucleus. Please remember this structure because it will be very important in the second topic. This is another indicator of CIN. So the rate of this micronucleus also increase in both YY2 defect and overexpressing cells. So to summarize this paradox, let's see this table. YY2 overexpression will cause the increase of spindle assembly checkpoint activity. It will delay the mitotic time. It will increase cell death. It will suppress tumor. YY2 knockout will decrease SAC activity, will shorten mitotic time, will suppress cell death, and increase tumorigenesis. However, both of them cause the chromosomal instability. So what is the relation between this? This is confusing. Again, we go back to the clinical samples. We try to analyze by ourselves data from the TCGA, from the clinical sample patient, clinical sample of a patient with CRC or colorectal cancer cells. We can see here that the Q3 sample or the Q3 group or group with moderate CIN has the worst prognosis, while group with the high CIN, Q4, has the best prognosis. Okay, so we question, is it possible that the cells with very high CIN actually has died in our cellular experiments? So we did not count it because we can't count them because they already died, maybe. So we tried to analyze this. Actually, Randy and me struggled for this, for proving this idea for several months. We tried to think a lot of methods because we need to analyze dead cell, which is impossible. So at the end, we tried to analyze what we call the dying cells. Cells which are already committed to die, they can't alive anymore, but still have compact structure of chromosome. So we can still analyze the chromosome, we can still analyze the micronucleus, which also contain the DNA. So actually we use the cell sorter and take the cells from here. This is the living cells, and this one, annexin 5 positive, but before they turn to PI positive cells. So we call them dying cells because here DNA has not been fragmentized yet, and we can still stain the nucleus and micronucleus. Okay, let's see here. This is the living and dying cells from the control group and the YY2 overexpressed group. See, in the control group, there is no significant difference between the micronucleus rate in the living and dying cells, because these cells just die normally. Some of the cells die, right? But in the YY2 overexpressed cells, you can see there are very high micronucleus rate in the dying cells, right? So cells with excessive CIN actually has died in our previous analysis, so we did not count them. So it seems that both YY2 overexpression and knockout resulted in the same level of CIN. But actually, YY2 overexpression should cause more higher CIN because the cells already died and we can't count them. Okay, so how about the CIN in YY2 knockout cells then? They also have higher CIN, but they are more tumorigenic. So we assumed that maybe the CIN was not high enough. It's not high enough, so it is in the level of the moderate CIN which drive tumorigenesis. So we then tried to increase the CIN using the SAC inhibitor, riversine. So in the YY2 overexpressed cells, the SAC become very high. Here we try to make the SAC even lower and lower. Using the riversine, you can see here the micronucleus rate increased more than merely knocking out the YY2, and then the cells died. So from here actually we concluded that excessive CIN, nevertheless, whether they come from SAC hyperactivation or hypoactivation, will cause cell death, tumor cell death. Okay, then how about the cells with moderate CIN? Actually, we observe another, from that cell sorting, I have showed you this data several slides before. Let's take a look about these two. The living cells in the control group and YY2 overexpressed group. The micronucleus rate here in the living cells of the YY2 overexpressed group is slightly higher than the control group. So it seems that this is cells with moderate CIN. What is the fate of these cells? And then, by this, we think about another phenomena in the clinical, and a very big problem in clinic, that is residual tumor cells, or tumor cells that survive chemotherapy. So, for this, we tried to establish a moderate CIN model by, we treated the cells with doxycycline, that is to induce the overexpression of YY2, because we use a YY2 inducible factor here. So there are two groups. This is the trans group. Trans group, that means we overexpress YY2 just for a while, and then after that we stop YY2 overexpression. And Conti group, that means we overexpress the YY2 continuously. So for the trans group, after stopping the overexpression of YY2, the YY2 level returned back to the level of control. And the CIN of these trans cells is about between the control group and the cells continuously overexpressing YY2, or the Conti group. It's a moderate one. This is also the other indicators of CIN. So trans cells have moderate CIN level while expressing YY2 similar with the level of control. So what is the fate of these cells? Our first experiment result is disappointing, honestly saying. Because when we try to compare the viability of these two groups, control, trans, and Conti, you can see here the viability of control and trans group were similar. The cell death rate of control and trans group also were similar. So we said that's nothing happened. But then, when I and Randy discuss about this, then we come to the question, maybe because the cells doesn't experience any stress. How if we put stress on them? So we try to treat them with oxaliplatin or drug. We give them drug stress, and you can see here, this is the control group, the red one, and this is the trans group. So the trans group has higher viability when treated with oxaliplatin. And the cell death rate, this is control group, this is trans group. The trans group is more resistant to all oxaliplatin treatment. So it seems, without any stress, it seems no different with control group. But when you give stress to them, they are more resistant. That means these cells are more adaptive to the environment. And then we make a model for residual cells, residual tumor cells. We treated the cells with oxaliplatin, and then we try to induce YY2. Let's see about the residual cells. That is we treated them with oxaliplatin. You can see here the micronucleus rate, or the CIN, is slightly higher than the control group. So residual CIN have moderate residual tumor cells have moderate CIN, and they have more viability or more resistance to oxaliplatin. But when we try to induce the CIN to become excessive, then the viability decreased. So then we tried to prove this in vivo by using xenograph experiments. So we treated the cells, we treated the tumors with the oxaliplatin only or YY2 overexpression only, which can suppress tumor growth to half of the control group. But when we treated the tumors with both YY2 overexpression and oxaliplatin, this will increase the CIN, so it becomes excessive. Then the tumor growth was suppressed until less than 1/8 of the tumor group. So this work was done by Randy. So the tumor cells, they can have CIN, moderate CIN will drive tumorigenesis, will increase drug resistance. Excessive CIN will cause cell death. And in the DNA damage, for example, when you treated the tumors with oxaliplatin, it can also cause moderate CIN. This will cause the residual tumor cells. But if we treated them with, for example, YY2 overexpression or other ways to increase the CIN, we can make this CIN become excessive and it will cause tumor death. So this was published in Advanced Science in 2024, and this is the dissertation of Randy. Yeah. So Randy is a very outstanding student. Another publication from him is this one, in review about the CIN, the two sides of chromosomal instability, drivers and breaks in cancer. It was published in Signal Transduction and Targeted Therapy with the IF more than 50. And it is now has been cited by Nature Communications, eLife, PNAS, and so on. The total citation more than 120 is now still a highly cited article. And also he also obtained a lot of outstanding awards from our university. Outstanding international student, outstanding international graduate, and so on. So, related with this one, we come to the second paper from Randy's junior in our lab, already graduated also last year. So when we try to analyze the KEGG, when we try to perform KEGG analysis using the transcriptomic analysis from YY2 overexpressed cells, we found that YY2 overexpression enriched DAGs related with immune system and cell growth and death. Sorry. Yeah, next please. Okay. And this is the TEM, or the transmission electron microscopic images. We found that YY2 induced cell swelling and membrane rupture, which is characteristic of pyroptosis or immune-induced cell death. And also the LDH release and IL-1 beta release also increased by YY2 overexpression. And please note that this happened not only in MSI high tumor, but also in MSS type CRC. I need to mention here that ACT116 is MSI high type CRC, which is hot tumor or tumor sensitive to immunotherapy. But HT29, this is MSS type CIC or cold tumor, CRC or cold tumor. They are not sensitive to immunotherapy. So this effect was observed in both hot and cold tumor. So this is the xenograph experiments. YY2 overexpression, of course, it suppressed tumor growth. And actually, it increased the expression level of the IL-1 beta. Again, this is a cytokine. This is an inflammatory factor. And also increased cell death. So we prove also in the animal experiments that YY2 induced pyroptosis. Next, please. Okay. Then we took the tumor lesions and subjected them to the single-cell RNA sequencing. And we found that the infiltration of the immune cells increased when YY2 was overexpressed. So this is the single-cell RNA sequencing analysis. We can also see the lymphoid cells, myeloid cells, or other immune cells also. And we can see that CD45 positive cells or immune cells infiltration increased in the tumor lesions performed by YY2 overexpressed cells. Next, please. Okay. And then we performed more analysis, and we found that the immune cells was CTL, or cytotoxic T cells, CD8 positive cells, CTL, which can kill the tumor cells. So CTL proliferation increased, CTL infiltration increased, CTL activation also increased, CTL exhaustion decreased when YY2 was overexpressed. Okay, next please. Okay. And then we tried to analyze the activity of this CTL in vitro. We took the spleen from mouse, and then we obtained the CD8 positive cells. We activate this, and we co-culture it with tumor cells, MC38 control or MC38 cells overexpressing YY2. We co-cultured them, and we found that when we co-cultured these spleen CD8 positive cells with cells overexpressing YY2, you can see that the cell death rate increased drastically to more than 60%. If you just overexpress YY2, cell death rate increased, but only to about 20%. Treatment with CD8 positive cells or CTL cells also cause cell death rate about 20%, but a combination of co-culturing them boost the cell death rate to more than 60%. So by this we concluded that in fact the YY2 overexpression can increase the inflammatory environment, and this will boost the activity of the CTL in killing tumor cells. Then we come to the question how YY2 triggers pyroptosis. I skipped the detail, but we found that the geo-enrichment enriched the DEGs in virus receptor activity, viral process, and double-stranded DNA binding. This is confusing. We don't infect the cells with anything. But another important thing is we found that in the YY2 overexpressed cells, the level of cytosolic double-stranded DNA increased. So this double-stranded DNA was recognized by AIM2, which is a double-stranded DNA sensor, and this activates the caspase-1 GSDMD pathway, and this will cause the pyroptosis. So the YY2 overexpression makes the cells to recognize and activate something, the double-stranded DNA, and they recognize it, the cells recognize it like a viral DNA, and it activates the viral DNA pathway, and it triggers pyroptosis. Where does this double-stranded DNA comes from? So actually, as I mentioned before, remember the micronucleus, right? I have shown you that the YY2 overexpression increases micronucleus rate. So micronucleus is a nucleic-like structure. However, the membrane of micronucleus is leaky and easy to break down because it lacks core nuclear envelope membrane, non-core nuclear envelope membrane like lamin B. Lamin A, C, and the others are core nuclear envelope membrane. Micronucleus also have it, but it lacks non-core non-core NE proteins. So it is not stable and leaky, and it's easy to break down. So we suspect that the double-stranded DNA detected when we overexpress YY2 comes from micronucleus. So to prove it, we strengthen the micronucleus structure by overexpressing lamin B. Micronucleus lacks this protein. We try to stabilize the micronucleus by overexpressing lamin B. So by this, we stabilize the micronucleus, and the cytosolic dsDNA decrease even when we overexpress YY2. So we overexpress both YY2 and lamin B, then the cytosolic dsDNA decrease, and the cell death also decrease. So it seems that the double-stranded DNA from the micronucleus, which is easy to break, was recognized as a viral-like DNA by the cells, and it triggers the pyroptosis. This caused the increase of the inflammatory microenvironment in the tumors. So again, by stabilizing the micronucleus, the LDH release, which is an indicator of the pyroptosis, and also IL-1 beta release also decreased. And the cell death also decreased. And this was also proven in vivo using the xenograft experiments when we overexpress both YY2 and lamin B. The tumor suppressive effect of YY2, which is the red line here, was canceled and partially restored by lamin B2 overexpression, the green line here. And the infiltration of the CTL cells also decreased. So YY2 overexpression increased CTL infiltration, but when we overexpress both YY2 and lamin B2, it decreased. And also CTL proliferation, CTL activation decreased by overexpression of both YY2 and lamin B2, but the CTL exhaustion increased. So lamin B2 overexpression, which stabilized the micronucleus, canceled the inflammatory response increased by YY2. So now, inflammatory environment can boost the effect of immunotherapy. This is already reported. So immunotherapy, one of the immunotherapy is the immune checkpoint inhibitors or ICI therapy. So this therapy activates immune system, especially T cells, and promote their killing activity. This is a promising tumor therapy, anti-tumor therapy, but its effect is mainly in leukemia. In solid tumors, the effect is still limited, especially in cold tumors. This type of tumors, we can say it is not sensitive at all to ICI therapy. So increasing the inflammatory environment has been reported as a way to increase the ICI therapy effect. So we tried to increase the tumor inflammatory microenvironment by using YY2 overexpression, because as I showed to you, it increased the cytokine secretion, it increased CTL infiltration, and so on. So you can see here, this is in hot tumor first. The red one here is the treatment with anti-PD-L1, a very classical ICI therapy. You can see that it suppressed tumor growth, but not so much. Not so much. From 26-fold to about 20-fold. YY2 overexpression, it suppressed tumor growth more, to about half, to 13 folds. But when we combined YY2 overexpression and anti-PD-L1 therapy, tumor growth was suppressed to from 26-fold to less than 4 folds. And the CTL infiltration increased by the YY2 overexpression, both YY2 overexpression and anti-PD-L1 therapy. This is in hot tumor model. How about in cold tumor? You can see here in cold tumor, you use another CRC cell. In cold tumor, there is no significant difference between control group and anti-PD-L1 treated group. So this kind of tumor is not sensitive at all to ICI therapy. But when we combine YY2 and anti-PD-L1, of course the effect is less than the hot tumor, but it still can suppress the fold change of tumor from 32 folds to less than 6-fold. So it can also sensitize cold tumor to ICI therapy, or what we call to turn the cold tumor to hot tumor. So, yeah. Okay. So the CIN can sensitize hot and cold CRC to ICI therapy by inducing pyroptosis. So this work was done by Dr. Tuan, who is now a researcher in one of the very famous hospitals in Chongqing, and was published last year also in Advanced Science. Okay, so as the summary of this part, we first proved the just right model of the CIN in tumor. So CIN can drive tumor regenesis, can benefit tumor cells, but if the level exceeds a threshold, then it can cause tumor cell death, and we can use this to improve tumor sensitivity to drugs, and also to immunotherapy, turning the cold tumor to hot tumor. Okay, so actually our group have done a lot of work using about the yin-yang family and the other hallmarks of cancer. Oh, sorry, next please. Okay, so this is the summary of some of our research, our publications from our group. YY1 and YY2 is involved in a lot of tumor hallmarks, including vasculature, so promoting angiogenesis in tumor. This was published in Cancer Research in 2018. It also caused the deregulation of cellular metabolism in tumors. Here are the publications. Resisting cell death, ferroptosis, and pyroptosis. Pyroptosis is just the one that I just explained to you just now. And also and sustaining proliferative signaling and genome instability and mutation. And because Prof. Ina's group is doing the topics about drug resistance and CSC, so I will show you some slides, a few slides about our results in CSC, or cancer stem cells. So we know that cancer stem cells are more resistant to therapy compared to bulk tumor cells. And cancer stem cells are the problem, one of the core problem of the tumor progression, metastasis, chemoresistance, and recurrence. And so if we treated the tumors with the conventional anti-tumor therapy, we can kill the bulk tumor cells, but the CSC remains there, although they are very few, so you cannot detect them. But somehow they can resuscitate and make another tumor lesions. So if we can, one of the thinking for targeting CSC is to make them differentiate. So they lost their stemness, they differentiate, then we can treat them with classical anti-tumor therapy. So actually, there are 3 types of division of cancer stem cells. First one, self-renewal symmetric division. So one CSC becomes 2 daughter cells, both of them are CSC. Another one is differentiated symmetric division, generated 2 differentiated daughter cells. Another one is asymmetric division, one CSC divided into one CSC and 1 differentiated daughter cells. So this is very problematic, asymmetric division, because by this, CSC can maintain the number of their population while growing the tumors, because the differentiated cells can grow more faster than the CSC themselves, which are often in the dormant situation. So actually, when we overexpress YY2, again, this is about YY2, we can see that the frequency of CSC decreased. This is in vitro, and this is in vivo. So the frequency of CSC decreased from one in every 4 cells to 1 in every 13 cells. In vivo experiments also resulted in the similar tendency. 1 in less than 20,000 cells, 200,000 cells, to become 1 in about 3 million cells when we overexpress YY2. So how about the effect of YY2 on the CSC division? We use a marker that is called it NAMP, which is a marker of non-CSC daughter cells. So if the CSC divides asymmetrically, one of the daughter cells will be NAMP positive, one will be NAMP negative. If CSC divides into 2 NAMP negative daughter cells, that means it generates 2 CSC. We call it CC division. This one we call it CD. If they generate 2 NAMP-positive daughter cells, we call it DD differentiation. Both of them are differentiated cells, not CSC anymore. So YY2 overexpression caused the decrease of the CD division, or division generating 1 CSC, one differentiated tumor cells, and increase the DD diffusion. That means it can cause the exhaustion of CSC pool. So, I skipped the detail, but which factor can determine the type of CSC diffusion? One is the mitochondria quality. Mitochondria, we know it as an organelle in the cells, is an energy factory, but actually the quality of a mitochondria determines a lot of things, more than the energy in the cells. So mitochondria, when it becomes aged, it will have a lot of damage. To maintain the quality, mitochondria will divide, or we call it mitochondrial fission. By this division, it will generate one aged mitochondria with a lot of damage and one healthy young mitochondria. During CSC division or during stem cell division, actually, cells with more young mitochondria will maintain the stemness. Cells with more damaged or mitochondria or aged mitochondria, they will lose the stemness and become more differentiated, for example, progenitor cells or other more differentiated cells. So actually, what happened was YY2 caused the increase of damaged mitochondria. We call it spaghetti-like mitochondria. Mitochondria should be an oval structure. But you can see here, this is like spaghetti, very long. So spaghetti-like mitochondria increased because YY2 suppressed the expression of DRP1. DRP1 is the protein that initiates mitochondria fission. So DRP1 accumulates here, and then it will just like pressing the mitochondria into two, with the damaged one concentrated in one of the two daughter mitochondria. So by overexpressing, so YY2 suppress the DRP1, so we try to rescue by overexpressing the DRP1. So the overexpression of DRP1 restored the percentage of CD type diffusion suppressed by YY2 overexpression. And then overexpression of DRP1, so it restored the frequency of CSC. When we overexpress YY2, the frequency of CSC in these animal experiments becomes lower from 1 in every 200,000 to more than 1 in more than 3 million. By overexpression of DRP1, it was restored into 1 in every 500,000. So, sorry, next please. So this work was published in 2023 by one of our alumni, Dr. Wei, who is now a lecturer in another university in China. And last but not least, this is unpublished data. This is totally different from the one I'm talking about just now. So where does the CIN comes from actually? Yeah, we can say that YY2 increased and CIN increased, that's right. But we know that cancer is a disease of mutation, because of mutation. And a lot of physical and chemical factors can induce cancer, right? So what is the effect of physical factors or chemical factors with CIN as a driver, a very fundamental driver of cancers? So maybe this is not very well known, but matrix stiffness is one of the physical factors very important in cancer actually. So we know hypoxia, we know starvation, or imbalance in nutrition and so on, but actually there is another factor that is the matrix stiffness. Maybe we did not aware about this, but there is a very good example from liver. We know that healthy liver is not so hard, it's quite soft, right? But liver we can have a disease called liver fibrosis, right? And then liver fibrosis, we know that liver fibrosis increases the risk of liver cirrhosis. Liver fibrosis and cirrhosis increase the risk of liver cancer. This is a very well-known example in clinic. But We didn't aware that before. A lot of us didn't aware, but this is now, we have several reports already actually, not from our group, ours is still unpublished, that this increase of the liver stiffness can actually drive liver cancer. What is why? It is still not well understood. But it increased the liver cancer. So actually the increase of the stiffness of tumor extracellular matrix, we know that tumor is surrounded by extracellular matrix. The increase of the stiffness of the ECM can cause a lot of changes in signaling pathway. It can drive activation of a lot of transcription factors and so on. So this is unpublished data. This is now under review in Nature Biomedical Engineering. I don't know whether it can, I hope it can go smoothly. So this is a spatial transcriptomic analysis from clinical patient with SCC. This is the collagen 1A1 and 1A2, or marker of stiffness. You can see that the collagen 1A collagen distribution is not equal, but the place or the location of collagen with high collagen also have high CNV score. CNV is one indicator of CIN, or copy number variant. And this is the single region genetic alteration analysis. So the changes of chromosome gain or loss, both chromosome gain and chromosome loss, is higher in stiff matrix region compared to soft matrix region. So it seems that the matrix stiffness is related with the CIN, chromosomal instability. Or here we use the indicator CNV. So we also do the orthotopic tumors in liver cirrhosis model. We established a liver cirrhosis model. We grow the tumor. We transplanted the tumor cells directly in the liver. And then you can see that the liver, in the liver cirrhosis model, the stiff one, the tumor growth much more faster than the non-cirrhosis one, the soft one. And then we perform the whole genome sequencing. Again, the gain and loss of tumor, chromosome is higher in the cirrhotic model compared to the non-cirrhotic one. I skipped the detail, but what happened was actually matrix stiffness increased the nuclear envelope rupture. So you can see here, BAF, lamin AC here is the marker of nuclear envelope, BAF is the marker of nuclear envelope rupture. So this BAF positive cells increase where the cells was grown in stiff matrix. This is another evidence. Here we overexpress the GLP in the nucleus. And then if we culture them in the matrix, and in the stiff matrix, you can see that during this 45 seconds to 1 minute, you can see that the GLP, which is a GFP which is concentrated in the NLS, in the nucleus, was dispersed to the cytoplasm for a while. And then, of course, because the cell has the ability to repair this NE rupture, the GFP was concentrated again in the nucleus. But this rupture, although it is only for a while, this exposed the DNA into cytoplasm, right? By this exposure, DNA damage will increase. Then the cells will repair the DNA, but during the revision, this will increase the mutation, the non-homologous end joining, or non-homologous repair, and so on. So this will increase the chromosome error. So this is the reason. So we found that matrix stiffness increased genetic alteration by promoting NE rupture. So, and then I skipped the detail, but through the multi-omics analysis plus a TAC6 and so on, we found that a transcription factor called MAS is critical for this regulation. So when we knock down the MAS using the shRNA, even if we culture the cells in the stiff matrix, the NE rupture event will decrease. So, yep, this is some of our unpublished data. Sorry, next please. So this is acknowledgement for our collaborators and for the grants. And this is our lab members. You can see there are many international students. So after Randy was graduated last year, actually we don't have any more Indonesian students. Hopefully in the near future we have, yeah. But we have some Pakistani students also here. Okay, next please. So this is international students from our lab. We have already 10 international students graduated from our lab, 9 from Indonesia. Okay, so 8 master students. Thank you. 8 master students and 1 doctoral student. Now we have about 4 international students from Pakistan. So the performance of our international students, especially from Indonesia, is very good and is very welcome also by other professors in our college. So here is our international students with Dr. Ma, who is the Wali Kelas for international student in our college. During international festival, we have Indonesian booth, we taste Indonesian food, they perform Indonesian dance, saman, and also tari Bali, and so on. And also this is Agnes, our alumni, who is now in the, she's a staff in the Widya Mandala Catholic University in Surabaya. Agnes was graduated in 2018, I think. She moderated me when I visited Vidya Mandala. And this is Sharon, who was graduated last year. She represents international students from the whole university to deliver a speech during the graduation. So 3 student representatives, undergraduate, postgraduate, and international student, and she was elected. This is Randy, during a seminar about YY1 family. It was held in Catania, Italy in 2023. He attended it online. And this is our groups winning the second award for the Innovation and Entrepreneurship competition. So these are the others outstanding awards obtained by our international students in our lab. Okay, so now at the last part, I will introduce to you this for study and research in Chongqing University. So actually, the annual, I have calculated it, and it seems that Chongqing, it is different from Shanghai or Shenzhen, whose living cost is actually very high. So the monthly expenses in Chongqing is totally, except the dormitory, the total monthly expense is around 1,500 RMB, or it's about 4 million rupiah 1 month. So it is not so different from Jakarta, I think. About sekitar 4 jutaan ya. So annual fees, the dormitory is about 10 million rupiah per year. The tuition fees, kali 2.000. Jadi the tuition fee is 100 juta lebih per year. Ya, tapi dorm itu sekitar 10 juta per tahun ya, 10 jutaan per tahun. Okay, so this is the annual fees and expenses in Chongqing. And the scholarship. The scholarship, we have 3 scholarships here. Chinese government scholarship, and actually the deadline is tomorrow. Yeah, the Chinese government scholarship includes the registration, tuition, and dormitory fees, medical insurance, with the allowance 3,000 RMB, or about 6 to 7 million rupiah per month for master student, or 3,500 for doctoral student per month. For the Chongqing University Presidential Scholarship and the Chongqing Municipal Government, I'm not sure about these 2 scholarships this year. Because for last year, the President's Scholarship we canceled the first class scholarship to give more scholarship to more awardees. So only waiving the tuition fee, either 100% or 50%. So the deadline for Chinese government scholarship, both for master and PhD student, is tomorrow. But if you are interested, and I hope if you can also share this with your friends and also with the master students, if you are interested in, just apply it online first with the documents you have. Because as long as I know, every year the admission office will mail you until 3 times to ask you to accomplish this and that. So you can still have about half a month, about 10 days or 2 weeks to accomplish the other documents. So the eligibility, non-Chinese citizen for master program, the age should be under 35. For doctoral program, the age should be under 40 years old. Language proficiency requirements, we have the English taught program. We have the English taught program in our college for both master and doctoral degree. IELTS more than 6 or TOEFL IBT more than 80, but this could be converted to, if you have TOEFL from institution, for example, you can use it also. You can use it also, and we can convert it. Or if your degree, because like my previous student from Universitas Jenderal Sudirman, some of them, they have bachelor in English, right? The international bachelor program. So they are Indonesian, but their bachelor program was taught in English. So if the university can provide you the evidence or the certificate that your previous degree was taught in English, you don't need to submit the English proficiency score. And or if you have studied in English-speaking country for more than 1 year, if you have the certificate, you can use it also, and you don't need the proficiency test. The GPA is more than 3 out of 4 or more than 80 out of 100. And applicants must not be recipients of any other scholarship. What you need is application form. This you could be filled in online, and after that you can download the PDF. This is for the Chinese government scholarship. You apply this first, fill it first, download the form, and then apply to the university. And then the information page of ordinary passport, notarized highest diploma. If you are prospective graduated student, you can have the evidence from the university. And then academic transcript, language qualification certificate, study plan. This is a very important thing, study plan. And then recommendation letters, 2 recommendation letters. And then physical examination form, non-criminal report or SKCK in Indonesia, right? And then the other supporting documents, for example, if you have published papers, just submit it with your application. And then for those who have obtained your doctoral degree, there is a program from the NSFC or National Science Foundation of China, like BRIN in Indonesia. So the NSFC program offered a program for outstanding young scholar from One Belt, One Road initiative country, and Indonesia is included in it, have obtained a PhD degree or have more than 5 years experience in research. And then under 45 years old with good health, able to work full-time in China for 3, 6, or 12 months. The allowance is quite interesting. So 3 months, 140 million Indonesian rupiah. For 1 year, more than 500 million rupiah. 500 juta rupiah per tahun. Okay, the deadline for this program is, we still have time, March 16. And postdoctoral program, as is also either from the university or from other programs, as also with a salary 200,000 to 300,000 RMB before tax. and we can apply for grant also. Yeah, but this is quite yeah. So last but not least, I'm also now serving as an editorial board member of BMC Medicine, JCRQ1 journal. So if you have a paper or review article, you can submit it to this journal, and I'm one of the member of the editorial board member. Okay. Okay, thank you very much, and this is my contact.
So thank you very much, Prof. Fifi. Very, very interesting and huge project you have presented to us. Thank you very much.
Thank you.
So I will open the discussion session, Q&A. Yeah. Prof. Asmarina. So three questions first. Also from online, yeah. So Dewi, could you please help me to data the online question? Not yet. So I also welcome the audience in the online Zoom to send your question via chat, and we will then offer this question to Prof. Fifi. So, Prof. Asmarina, first question. Okay.
I have to leave earlier because I have another meeting in the next door, like a wall of this from the administration thing for our study program. So yeah, thank you very much, Prof. Fifi, for your interesting topic and comprehensive data, and yeah, I cannot follow actually how many data that you have that I can understand. So, but maybe I can understand easy if I read your paper, yeah, because so many data about this. Yeah, I'm genetician, and I'm also I in epigenetic.
Okay, yes.
I do a lot of research also in epigenetic. What do you think about the epigenetic? Because you explained also about the extracellular matrix and so on. The environment can also influence the chromosomal instability, I think.
Yes,
And how about this epigenetic mechanism that can cause maybe the chromosome instability and the expression of the so many protein that you have already explained. And then the second question is about your opportunity. Is there any opportunity to our student to research stay for research for 3 months or something like that in your university? Because yeah, we didn't have so much equipment in Indonesia. We don't have so many sophisticated or complete equipment in Indonesia in our faculty also. Maybe our student can stay for 3 months or 6 months in your university or faculty, because our university can also give funding, but not so much. Yeah. But our university give also through the UI global funding for students to do research abroad. Yeah.
Yeah.
But yeah, we can also study program can also support a little bit funding for students that can do research in the abroad institution. So this is my question. Thank you.
Okay. Thank you, Professor Asmarina, for your questions. And actually, you hit a very important thing, that is the epigenetics, right? So epigenetics alters a lot. I think your question is what is the relation between epigenetics with tumor microenvironments, for example, the matrix stiffness I mentioned before, and the CIN, right? So honestly saying, you just hit the very important part. So actually in our unpublished data, the point is really the epigenetics. Yes, so it changed the chromosome, it changed the chromosome accessibility of a lot of things, including the transcription factors. And this transcription factor, as I mentioned here, mass, can change eventually it changed the epigenetics things through a long pathway, of course, and eventually it caused the nuclear envelope rupture and then the CIN. And also other factors, other reported pathways are also involved in the regulation of tumor microenvironment through epigenetics to the other tumor characteristics, including metabolism and so on. So tumor microenvironment is really, really very important. It changed a lot, the transcription factors, the epigenetics, the other signaling pathway, the inflammatory environment also, and so on. But the point is, I think, is the barrier, and that means who relays these physical and chemical factors from the tumor microenvironment and change them, who relays this and pass them to the biological pathway in the cells. And this will include the change in the epigenetics and tumor and the transcription factors and others. For your second question, that is about the opportunity to have the students from UE to come to our university. This is, I can say this is really possible. For example, with the joint funding. For example, one example is, for example, before a student from abroad come to our university, our college. So, for example, our partner university abroad support the students with the ticket and the living cost, while the experimental cost could be supported by us, for example. Yeah, it's possible for 3 months or, yes, and if they have the place in the student dormitory, I'm not sure. I need to confirm with them first because sometimes the dormitory is very full. But if they have space, of course, the student can also apply to stay in the dormitory. Yes, and it will reduce a lot the cost.
Thank you very much. Yeah. Because one student now stay in Mahidol University in Bangkok to do research supported by UI Global Funding.
Okay.
Yeah, I think maybe our student can also stay for a while, 3 months until 6 months in your university and from our university support.
Yes, we very welcome the international students, and we hope by that kind of collaboration, we can launch more collaboration in the future.
Of course, maybe we can also sign the MOU for our university.
That's what I want to remind, that we don't have any MOU with Chongqing University. So I think for that collaboration, we should propose this MOU from university to university, or maybe some agreement from faculty to college to college, or faculty to faculty, like that.
Yes.
Okay.
But if for student visiting, like for 3 months and something like that, actually for us, from our side, we don't need MOU first. We just can do it with LOA first, from lab to lab, supervisor to supervisor is okay. Yeah.
That's good. Thank you very much. Thank you very much, Prof. Asmarina. So before we continue the Q&A, please give big applause to Professor Vivi Kasim.
Thank you.
Sorry, I forgot to ask the,
Professor, while since I have to leave this.
You want to give a certificate first?
Certificate
Yes, the certificate ceremony for the speaker. So
just for memorize for you.
Okay.
The certificate also for Professor Ina. Prof. siapa? Prof. Vivi. This is for your reference. This is for you. Okay. And then another gift for you. Okay, no problem. Thank you for nice, interesting topic. Yeah, for me also, and personally, because chromosome and so on. Yeah, I interested also.
Thank you, Prof.
Okay, terima kasih. Ini Prof. Ina, I think you have already this. Okay, thank you.
Thank you very much.
Oh, ini. Yeah, just give it a little bit. Okay, small.
Okay.
Okay, thank you. Ya, I have to leave ya. Thank you very much. Terima kasih ya, Prof. Vivi ya. Kapan-kapan kita bisa MOU lagi, MOU. Sampai kapan di sini?
Sampai lusa udah berarti.
Oh, lusa.
Nanti harus datang. Kayaknya perlu sekali lagi datang. Karena joint supervision. Gimana? Itu yang kemarin. Thank you. Yes, we still have time, maybe about 10 to 15 minutes. It's okay. I will ask the audience from online. Is there any question? I welcome to the question. Or students here. Yes, please, Linda, Dr. Linda.
Yeah, thank you so much for Prof. Ina and also Prof. Vivi Kasim for your very comprehensive research. Oh yes, yeah, because of my voice, yeah, not clear yet. Okay, so I want to ask about, you mentioned about the YY2 have suppressed, you can, if a patient with the elevated YY2 have the best prognosis, right? And how about YY1? Thank you so much.
Yeah, very good questions. And so YY1 and YY2 are very highly homologous genes. And as you can see, actually during the evolution, YY2 comes from YY1. But interestingly, YY1 and YY2 have totally different roles in cancer. While YY2 is tumor suppressive gene, YY1 is oncogene. So in our previous paper, published in 2022, I think, we found that YY1 and YY2 can compete and bind to the same region of the promoter of the target gene. But one activates the target gene, one suppresses the target gene. So they have, although I cannot guarantee 100% they are totally have the opposite roles, but it seems that is, at least in cancer, they have opposite roles, and they can compete to bind to the same target genes. They compete to regulate the target genes. So this is interesting because they are very similar, right? Okay.
Okay, thank you very much. So please introduce yourself.
So thank you for the lecture. My name is Salisu Abdulhamid, an international student from Nigeria. So I have a question about your presentation about the relationship between matrix, extracellular matrix, and tumorigenesis. So you made an example with liver cancer. So I want to ask, is there any investigation also relating the extracellular matrix and tumorigenesis in other types of cancer? Because I want to ask more about like cancers that are existing in the blood, like leukemia. Do they also have any relation to do with the extracellular matrix? Thank you.
Okay, thank you very much for your question. So, yeah, the most famous model comes from the liver cancer. But how about the other cancer? We also have the, for example, fibrosis and cirrhosis and other organs or the tissues. But the relationship between them and cancer have not been well understood yet. Okay, the most famous one comes from liver. But for the leukemia, I'm not very sure because blood cancers, they are in the liquid condition, right? Yeah, so it will be more harder to study, I think. Okay, anyway, thank you.
Okay, thank you very much. So, Devi, you have a question? Okay.
Thank you, Prof. Vivi, for the very interesting presentation and research. Very complex and comprehensive, and it's very interesting. Okay, in this presentation, you said that there are 3 groups of CIN number, low, moderate, and also high. And my question is, how does the way for classifying low, moderate, and high CIN, and is there a threshold for that? Thank you, Prof.
Thank you.
Okay, thank you, David. This is a very important question, and this is still a gap between our basic research and the translational one. So in our research, we actually just classified them according to quartile, so 25%, 50%, 75%, and 100%. But how about the threshold in clinical patients? I think this is a very important gap that we need to study in the future. So we still need to, I think it will be different also between the types of cancer. What is the threshold of the CIN? And if we can have enough samples and determine the threshold of the CIN, of course it will be a very good strategy in determining what kind of therapy we should do to the patients. But unfortunately, we don't have enough data for that nowadays. Okay.
Okay, thank you very much. Prof. Vivi, you can take a seat. So, Pak Edi, maybe you have a question?
I think mostly the same with the Budewi, yeah, the threshold. Yeah. But another thing, can we combine with this kind of CIN, this one, with the like immune cell therapy, like CAR-T and so on?
Yeah, okay. We have not tried yet, honestly saying, with CAR-T. What we have tried we tried with anti-PD-L1 therapy, ICI therapy. But it is really very, very worth it to try to combine CIN inducer with the CAR-T and so on.
Sorry, maybe I want to add your data about the YY2 and PDL1,
Yes.
anti-PDL1. Is it clinical data or in vitro data?
In vivo, so with the
in vivo with the animal.
Animal experiments.
Oh,
Yeah.
okay, but
Yeah.
not yet translated
No,
to
not yet.
clinical.
Yeah,
okay.
not yet.
Okay, how far is it to be translated?
Yeah, this is still, we are now also trying to start the collaboration with clinicians to search about the opportunity to apply this, or at least we can have some preclinical, more closer to clinical experiments. But we are still now trying to collaborate with the clinicians from our affiliated universities, affiliated hospitals, I mean, and we are trying to.
Because it seems very prospective to be the additive therapy or complementary therapy to the immune therapy that we already well known now.
Yes.
And also with CAR-T, CAR-NK, the other immune therapy,
That's right.
cellular therapy.
Yeah.
Like that. Okay. Any other question from the Zoom? No question at all. And other question from PhD student, biomedical PhD student? No? So if no question anymore, I think we have learned so much very comprehensive knowledge from Prof. Vivi about especially about YY2, the transcription factor. I studied also in my PhD for transcription factor for cancer also. It's very interesting that you give me more knowledge about transcription factor in cancer stem cell and also in cancer. Yeah, and this transcription factor can be added to the treatment immunotherapy that we already known. That was a new insight for us. Okay, thank you very much again, Prof. Vivi, Prof. Wu, for visiting us today, for giving us very comprehensive knowledge. I hope in the next future we can collaborate more, not only in lecture, but in research, to do research together with the students and also the young staff, exchange staff, to give more result, more publication between both university or both faculties. Thank you very much again. Please give big applause to Prof. Vivi.
Okay, thank you, Prof. Ina.
Thank you very much. Yeah, I hope you have still 2 days here in Jakarta spent. For your information, I and Prof. Vivi come from the same not high school, junior high school. From the same, but I'm older. Dan in SMPK 2 in pembangunan, Jalan Pembangunan Sonia Gajah Mada. So we just realized that we came from the same junior high school. Very stay near. Okay, thank you very much, the audience, for today seminars or guest lecture. Thank you, Nusli, for accompanying us today. Oke, Prof. Vivi. Saya tutup ya. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang semua.
Maybe you want to take a photo together in here, Prof. Ina, with the Prof. Kasim. Yeah, Vivi Kasim. Oh, all the participant, of course. Yeah, yes. Everyone can go to the front. Adrian, Mas Nazar, Mas Nasar ya? Ada yang di luar, Mbak Nova? Enggak, oke. Mbak Nova ikut boleh? Enggak, oke. Banyak tadi ya. Salisu ya, oke. Oke, merapat. Oh, saya. Oh, oke. Ya. Oke, semuanya lihat ke kamera. Ya, 1, 2, 3. Oke, lagi. Gaya UI mungkin? Gaya UI? Yeah, UI. Okay, bro. Oh, okay. You on the left, I on the right. I always right. Okay. Thank you so much.